Alright guys, here we are with Chapter 11. It's all about cell communication. I know Chapters 9 and 10 were pretty rough, pretty extensive, and lots of heavy information. Respiration and photosynthesis are perhaps two of the largest or most complicated and most important of the cellular mechanisms we're going to look at in biology. So this chapter hopefully won't be quite as heavy and hard to digest. It's all about cell communication. When a cell or a group of cells or an organism go through the process of, say, photosynthesis or respiration, how do they communicate amongst all of the different organs, all of the different cells? How do they get that job done? Think about your body, which is made up of trillions of cells, like an enormous city, and within that city there are all different types of people doing all different kinds of jobs. And for that city to function, there has to be good communication amongst all the different residents. So the same thing happens in a living organism. There has to be good communication amongst the cells, and that's what Chapter 11 is all about. So our overview talks about the importance of cellular messaging. And obviously that's important if you have, say, like a human body, a creature that's made up of trillions of cells. You're going to have good cell-to-cell communication to kind of make sure that all the processes that need to occur for life go on without too much of a problem. This is also essential for just a single-celled organism as well, because within the cell, remember, there are lots of different organelles that each have their own job to do. So there has to be good communication even between within a single cell or between single-celled organisms, say like a colony of bacteria cells. They have to be able to communicate with one another. So it doesn't matter if we're talking about single-celled creatures or a creature that's more complex, made up of trillions of cells. There needs to be some way to get messages or information from one cell to the next. Biologists have discovered that there are some basically universal mechanisms for communicating or cellular regulation. So when we look at something as simple as a yeast cell, there are certain pathways of communication that the yeast cells use. And some of those pathways are almost exactly the same within the cells of a mammal, so like a human being. So it's pretty amazing. And again, this kind of points to an evolutionary mechanism, whereas these pathways or these methods are kind of shared via a chain of genetics down through time. Cells often communicate with each other using chemicals, and that's going to be the main chemical or signal is going to be a chemical. They can use touch, and there can be environmental signals such as light and temperature, but by and large it's chemicals that are passed from one cell to the next as the form of communication. We're going to look at a particular one. We're going to look at epinephrine. That's going to kind of be our model for what we're looking at here. Epinephrine is a signaling molecule that you're probably familiar with in the whole fight or flight mechanism. If you've ever had to give a speech in front of an audience and you felt really nervous and your heart starts pounding and you start to get kind of sweaty and your stomach kind of feels like you have butterflies, well, that's epinephrine. So epinephrine is a hormone that's released throughout the body, and it's a good model for chemical messengers and how those chemical messengers affect how the body reacts and how organisms react. Here we've got two animals. Their bodies are reacting to the epinephrine that's been released. Epinephrine is also known as adrenaline. So if you've heard of the term adrenaline, like when you're riding a roller coaster or you're playing a game of football or anything that causes fear or excitement, that's that fight or flight mechanism we're talking about. So here we have two different cases of that going on. You have the cheetah, who is obviously chasing as best he can to try to capture his breakfast so he can survive, eat, and live another day. And then we have this poor little guy, this gazelle, who is obviously running for his very life. So there's a cool little quote down here that I've always liked. It talks about how it really doesn't matter if you wake up in the morning as a gazelle or a lion, that you better be running. So epinephrine or adrenaline is the chemical that causes the body to react in preparation for fight or flight. So we want to kind of take a look at how does that happen? How does this chemical cause these changes within the body itself? So let's get started with concept 11.1, and it says external signals are converted to responses within the cell. And we can look at something as small as a microbe or a yeast cell, or something as complex as a human body, and what we find is that there are signals that start on the outside of the cells that get passed to the cells, and then there's a process where the cells take in that information and do something about it. So we're going to start by looking at some yeast cells, but the same thing applies. We're going to find all the way up through the evolutionary chain. So let's talk about those yeast cells. Yeast cells are microorganisms that are eukaryotic. They're within the kingdom of fungi, so they're along with mushrooms and molds and other types of things. They don't really have sex, as far as they don't have males and females. They do have types or mating kinds that are called alpha and beta. And what they want is for these two different varieties to get together so that they can mix their genetics. That increases the strength of the genetic pool. So these two different mating types, they need to have a way of locating one another. You're talking about a single-celled creature. It doesn't have the ability to move around. It doesn't really have the ability to see and smell and things like that, of course. So how do they do that? Well, they use something called a signal transduction pathway. And a signal transduction pathway is basically a series of steps. It starts on the cell surface and then migrates into the cell to cause some sort of change or reaction. Transduced means changed. So a signal transduction pathway is basically a series of steps where information gets passed from one cell to another cell, or from outside of the cell to within the cell. And this signal transduction pathway is how the different varieties of yeast basically find each other and end up mating together. So chemical signals that basically attach on the outside of the cell, those signals are converted or transduced into the cell to initiate some sort of response. And here we have the two yeast cells. We have the alpha on the left and the beta on the right. They give off certain mating factors. You might want to think of those as pheromones, certain chemicals. And the other variety has the receptor that matches that particular chemical. So here is the beta yeast cell. He's giving off his beta factor, and notice the shape of it matches the receptor on the alpha cell, and vice versa for this little factor right there. This actually causes them to move toward one another, to grow toward one another, and in the process they join together and they become this kind of supercell, this alpha-beta cell, and then they go through further division as part of the reproduction process. But what we're interested in is this actual process here, where one cell releases a chemical that causes a change in the other cell. When we look at how this occurs, when we look at the mechanisms for this cellular communication, the cool thing is these pathways are very similar amongst almost all living organisms. So you would, based on the evidence, you would say that these evolutionary pathways developed long, long ago in simple prokaryotic organisms and were carried over through eukaryotic organisms, through the entire line of evolution all the way up to modern day creatures that you see now. So if we compare some of these pathways between something as simple as a single-celled yeast and a mammal of some kind, you'll find that there's a lot of similarities. Even though the most common ancestor between a yeast cell, a fungus, and an animal would have lived billions of years ago, this pathway or this mechanism has been passed down via evolution to all these different creatures. One of the things that they look at is the concentration of these signaling molecules around them. So when you're talking about even the most simple of creatures, like these single-celled yeast, they need to communicate with their community or with their neighbors. And this is called quorum sensing, Q-U-O-R-U-M. So how do they know if they're all alone in the world or whether they are surrounded by others like them? Well, it's because of these chemicals that they put out to each other. So based on these chemicals, they can get a sense of where the other yeast cells are, how many of them are out there, and that type of thing. So it's kind of basically their telephone system or their instant messaging system. So they don't have the ability to see or hear or communicate that way, so they communicate using these chemicals. One good example is the formation of what we call biofilms. Bacteria and all simple creatures like that, they tend to do better if they work together as a team. They can get certain things accomplished that they couldn't get accomplished all by themselves. So these bacteria need to know when and if other bacteria are around them and how many are there. One of the things they do is they work together to create a biofilm. If you've ever gone out to the woods after a rain or early in the morning when there's still dew on the ground, and you put your hand on a log and it's really kind of slippery and slimy, that's probably a biofilm that these bacteria have formed to help them in the process of digesting and eating their food. Or when you wake up in the morning, when you have horrible morning breath and you got this nasty, slimy film on your teeth, well, that's a biofilm. That's the bacteria that live in your mouth They are working together to create this film, and then they are eating whatever leftover material is on your teeth. They're also responsible for releasing gases that cause that bad breath and everything else. So you want to do a good job of brushing those teeth. You want to break up that biofilm and separate those bacteria so they can't work together anymore. Another good example is a soil-dwelling slime bacteria called myxobacteria. Myxobacteria have a method whereby if there is a shortage of food, they will get together and start to form what's called fruiting bodies. And fruiting bodies produce spores, and spores are kind of like seeds in this case, that they're tough and they can survive through difficult conditions where maybe there's not enough food or water, and they'll basically go dormant within these spores, the bacteria will, until the conditions improve. So the way they know to do that is by this quorum sensing, by these chemicals that are sent out between them. So here you have a whole bunch of individual rod-shaped bacteria cells, and in the process of noting that, say, there's not enough food around, they will release these chemical messengers that will basically rally the troops, and they'll begin to join together and form what's called a fruiting body. And within these fruiting bodies, they'll produce spores. And these spores are very tough, dormant bacteria that they can survive a long time. And then perhaps at the next rain or when the next rotting log is nearby, they will basically come back to life and start the whole process over again. When we talk about cellular communication, there's basically a couple of different things to discuss, and usually you want to kind of divide it by distance. Sometimes the cells are right up against each other. They're basically touching each other. And if that's the case, the cells are going to use connections between them. So in animal cells, those were called gap junctions. If you remember, if the cell membranes are pressed right up against each other, sometimes in animal cells there's small openings where cytoplasm and other chemicals can flow between the two cells. That was called a gap junction. In plant cells, we call that plasmodesmata. So the plant cells have it a little bit harder because they've got the cell walls, but there's usually gaps, like little chinks in the brick wall. And those little openings or chinks in the brick allow for chemicals and materials to pass through, and that's called plasmodesmata. So that works if the cells are actually touching each other, if they're right beside each other. Usually, though, they're not. Usually, say if we're talking about single-cell bacteria, they are close by but maybe not touching, or a lot of times we're talking about animal cells that are not right close touching each other, so we'll have to then use some other types of communication. So here's a model of what happens when cells are actually in contact with each other. Here we have cell junctions, gap junctions in animal cells, plasmodesmata, and plant cells. Here we have cell-to-cell recognition where one cell has a particular molecule on its outside and another cell has a molecule that kind of fits in with that like a lock and key, and that causes a particular signal that passes information. So this would be if the cells are actually touching each other, and that is the case in multicellular organisms quite a bit, but oftentimes the signal has to travel farther from cells that are not touching each other, and we're going to look at that next. So if the cells are not touching each other, they have to send this chemical messengers, and we usually call them local regulators, if they send the chemical a short distance. And a good example of that is we're going to look at something called paracrine signaling. That would be, say, a cell releases a chemical directly outside of itself, and it affects just the cells that are right nearby. So that's a local regulator. Or we're also going to look at, say, nerve cells. When you reach out and touch a hot stove accidentally, there's a pain signal that gets sent from your skin all the way up your arm to your spinal cord and then into your brain. Well, that pain signal travels via nerves, and those nerves actually use electricity and chemicals. Between the nerve cells, there's a gap called a synapse, and they actually use chemicals to cross that little gap. That's very local. That doesn't go throughout the whole body. That's just from one nerve cell to the next nerve cell, so that would be a local regulator as well. So if we're talking about local short distances, there's a couple of ways to do that. We'll take a look at some examples, and then we'll come back to the longer distance. So here's some examples of local signaling. Here is the one called paracrine signaling. So if this cell is releasing certain chemicals that affect only the cells right around it, these chemicals diffuse through the extracellular fluid that surrounds them, and that would be local regulation. Here we have the synaptic signaling between two nerve cells. There's an electrical signal that gets to the end of the nerve cell, and then that releases chemicals. Those chemicals cross the gap, and cause an electrical signal to get picked up in the next cell, and it travels on down the line. But this is very local. This is only between one cell and the next cell, so it doesn't travel far throughout the body. So what if we need to travel a great distance? What if a signal has to get, say, from some central part of your body all the way out to the end of your toes? Well, in that case, in plants and animals, we use a chemical called hormones. So in plants, you would normally think of these as plant growth regulators. Oftentimes the term plant growth regulator is used instead of hormones. But both of these are chemicals that are used in plant and animal bodies to travel a greater distance, to go from one part of the body to the next. And you might ask yourself, well, say this adrenaline, this epinephrine hormone that's released near the kidneys, how does it get to the heart? How does it know to go to the heart? Or how does it know to go to the liver to release and break down glycogen to make glucose for fast energy? Well, the truth is it doesn't. This hormone, when it gets released, it goes everywhere. It's not site-specific. It gets carried throughout the entire circulatory system to every cell within the body. But what we're going to find out is not every cell responds to the hormone. Only certain cells respond. And it's because those certain cells have the right receptor molecule on the outside of their cell membrane. So the ability to respond depends on whether or not you have the right receptor molecule. So this hormone, epinephrine, is going to go throughout the entire body, but not every part of the body responds, and not every part of the body responds in the same way. Now, plants don't have a circulatory system like we do, so their plant growth hormones actually can diffuse through the cells. They do travel through some of the vascular system a little bit. Plants can also release these hormones as gas molecules into the air. So, for example, ethylene gas, which is the gas that ripening fruit releases, that can be released into the air and then taken in by the plant somewhere else. So rotting fruit or ripening fruit can actually cause other fruit on the tree to start to ripen faster because of the ethylene gas that gets released. So this will be kind of like long communication from one part of the body to the next, or from one part of the plant all the way to the next. We'll take a look at an example. So here we have a human body. So we have an endocrine cell. This would be located near the kidneys. It's releasing, that would be epinephrine. So this is endocrine. That's a different part, but it's similar to epinephrine. It's also a hormone. If it's released by one particular body part, it goes into the blood vessels. The circulatory system carries that throughout the entire body, but not all cells respond, or not all cells respond in the same way. Only the cells that have the correct receptor sites that binds to that hormone get that message. So it's not like the hormones know where they need to go. They just cover the entire human body, but the parts that need to respond are the ones that have the correct target cells on the outside. Let's do a quick preview of the 3 stages of cell signaling, and then we'll take a break. Back in the late 60s, early 70s, there was a scientist named Earl Sutherland. He actually won a Nobel Prize for his work with this hormone epinephrine. He was trying to figure out how epinephrine works, because in the fight or flight mechanism, when an animal gets scared or feels like it's going to have to fight for its life, one of the things that happens is in the liver, glycogen starts to get broken down into glucose so that there can be a surge of energy to the muscles or to the heart or to the respiratory system. Remember, glycogen is like the short-term energy storage for animals. It's the thing that gets broken down first before fat gets broken down. So how does this epinephrine break down the glycogen? Well, first Sutherland did a pretty interesting experiment. He took glycogen and epinephrine and put them in a test tube just to see if the epinephrine actually did the breaking down of glycogen. And it does not. If you mix those two together, the glycogen doesn't get turned into glucose. So what he realized was this must be a multi-step process, because then he took living cells and he put them in the test tube, and there was glycogen in the test tube, and then he added the epinephrine, and the process occurred. So what he realized was it must be a process of multi-steps that takes place in the living cells. It's not actually the epinephrine that breaks down the glycogen, but the epinephrine must be some sort of signal that kicks off the process. So what he discovered was there's basically 3 steps to this process, and they're called reception, transduction, and response. For a nice quick overview, you can take a look at this animation and just hit right click and play, or I'm just going to show you some still photos or still models in just a moment. So here we have the first one. This is reception. Reception is where there is a particular signal molecule that is floating around in the extracellular material or taken up by the bloodstream. And in certain cells, in the target cell, there's going to be a particular receptor that matches that signal molecule. So this ligand or ligand is a signal molecule that will latch onto or connect with this receptor, and that will usually change the shape. Remember, most of these are proteins, and when this ligand or ligand gets into the proper spot, it will change the shape of the protein. So this is reception. Not every cell responds to that particular signaling molecule, only the ones that have the proper receptor. The next step is transduction. Once the receptor receives the signaling molecule, that causes a whole chain of events to occur, and usually involves a number of proteins and a number of other signaling molecules that kind of get sent along the way. So it's not just a quick 1, 2, 3 steps. Oftentimes in transduction, remember transduction means to change or a change, this signal gets transferred and it goes through a whole series of proteins in this pathway called transduction. Last but not least, we're going to reach the end of the line and we're going to have some sort of final process. What was it that we were trying to make happen or occur? And that could be some sort of enzyme that responds, that could be a change in the cytoskeleton, that could be turning on or off a specific gene within the cell. So in response, you have basically the final finish line. What was it that was to be accomplished after all of this process? All right, let's take a break right there. We're going to look at these 3 parts: reception, transduction, and response, all about the ways that cells talk to each other and send messages, and we'll get into more detail in just a moment. Be right back.